Hello everyone, my name is Frank, and today I'll be presenting to you some of the key findings from a new study performed by Castro, Gill, Legamari, and D. Giacinto on the role of CDK1 in the disassembly and reassembly of the nuclear envelope in vertebrate mitosis. Micronuclei arise from chromosomes and DNA fragments that fail to be incorporated into the primary nucleus after cell division. These structures are a major source of genetic instability following DNA repair and replication, and this instability leads to defects in DNA coupled to aberrant nuclear envelope formation. This instability also ultimately leads to a spectrum of chromosome rearrangements called chromothripsis, a phenomenon that is a hallmark of several cancers. And despite its importance, the molecular mechanism at the origin of this instability is still not understood. The study showed for the first time in vertebrates that polo-like kinase 1 not only participates in nuclear envelope reformation control pathway, but is essential for timely disassembly of the nuclear pore complex at the onset of mitosis, and for preventing the loading of nuclear pore complex on the chromatin of lagging chromosomes during mitotic exit. The study provides mechanistic insights into the pathway of nuclear envelope reorganization in vertebrates and sheds light on the possible identification of specific counteracting phosphatases that may be an important aspect to pursue in order to understand the full picture. In order to understand the causes of micronuclear instability, Castro and colleagues analyzed the composition of micronuclear envelopes in HeLa and U2OS cancer cells by immunofluorescence. From this, they found that when HeLa cells were stained for the presence of importin beta, a mediator of pore complex interactions, the MPC marker MAB414, and nuclear structural protein lamin AC, there was heterogeneity among the micronuclei in relation to the presence of these three markers. However, the majority of the micronuclei were lacking importin beta. They also found that nuclear pore proteins, NPCs, and importin beta recruitment precedes lamin A, and it starts loading at the poleward side of the segregating chromatids. These results not only indicated that importin beta and MPC do not assemble on lagging chromatin in the anaphase, but these observations also suggest that either 1. The processes of laminar reassembly and MPC reassembly after mitosis are independent of each other and are under different regulatory systems, or 2. Recruitment of MPC components to the anaphase chromatin is negatively regulated directly or indirectly by factors, possibly kinases, acting at the central spindle or limited by the small amount of chromatin present. But which of the previous indications was more likely? To find the answer to this, Castro and colleagues fixed and stained HeLa cells for MEL28 and PLK1 on lagging chromatin as indicated by the arrows. From this, they found there was an absence of MPC component on a substantial mass of lagging chromatin, indicating that the first hypothesis was true. The processes of laminar and MPC reassembly after mitosis were independent of each other and were under different regulatory systems. They then tested if inhibition of different kinases present in the anaphase midzone would allow MPC loading on lagging chromatin in vertebrates. HeLa cells were treated this time with DMSO as a control, PLK1 inhibitors BI and MLN, and Aurora B inhibitor ZM for 4 hours. These cells were then fixed and stained for the lagging chromatin in telophase cells and analyzed. Inhibition of either PLK1 or Aurora B was able to release the inhibitory effect and allowed part of the MPC loading to the lagging chromatin. The effect caused by PLK1 inhibition was surprising, as this kinase has never been reported to act on nuclear envelope reformation. These findings were further supported when HeLa cells transiently transfected with M-cherry PLK1 and fixed and stained for MAB414 showed a significant decrease in the level of nuclear pore complex proteins. To better understand the molecular mechanisms, Castro and colleagues also tested the relevance of protein kinases in envelope reassembly. DT40 chicken cells were arrested in nocardazole for 4 hours to remove spatial cues and isolate the molecular mechanisms underpinning these processes. The cells were then treated with DMSO, cyclin-dependent kinase inhibitors, and PLK1 and Aurora B inhibitors as well. In DMSO-treated control cells, lamin A, MPC, and importin beta are all dispersed in the cytoplasm. However, when CDK1 was inhibited, there was a high MPC recruitment on the chromatin in the system. The effect was even more pronounced when CDK1 and PLK1 were inhibited together. Inhibiting either PLK1 or ORB alone, on the other hand, produced no effect. These results seem to indicate that inhibition of CDK1 and PLK1 in pro-metaphase cells are both required for the complete loading of the MPC and the importin beta on the chromatin. Castro and colleagues further assessed PLK1 involvement and found that when PLK1 was inhibited in DT40 cells, there was a significant increase in importin beta and MAB414 staining from the diffused state towards the chromosomes. In PLK1 inhibited cells, importin beta and nuclear pore proteins are either still around the condensed chromosomes or in distinct patches within the cytoplasm. These findings further indicate that PLK1 is required for complete disassembly of the MPC at the onset of mitosis. In light of these findings, Castro and colleagues proposed a new model for the regulation of nuclear pore protein complex reorganization in mitosis. This study proposes a sequential model for the nuclear envelope reformation, where lamin A targeting to the chromatin is under the control of the decreasing cyclin B CDC2 activity, while the MPC loading is also spatially regulated by PLK1 activity. These findings shed more light on the mechanisms involved in cancer cell formation and add extra value to the use of a new form of cancer drug treatment in the form of PLK1 inhibitors for the treatment of specific cancer types arising from micronuclei formation.